Hi all, welcome back to another session on protein synthesis translation. In the previous video, I have explained about how RNA is transcribed from the DNA strand. Today, we are going to see how translation of the mRNA, which is carrying the code, happens on the surface of the ribosomes. So, let us see the steps involved in translation process. The process of translation, it involves the order in which the sequence of nucleotides in mRNA are translated into the sequence of amino acids in a polypeptide chain. The mRNA has a series of codons made up of triplet bases, and the codons pair with the anticodons of the RNA molecules, and the pairing will be between AU and GC. As you all know, in case of RNA, in place of thymine, uracil is present. So, the pairing will be A to U and G to C. The whole process of translation is completed in 5 steps. The first step being activation of amino acids, second one transfer of amino acids to tRNA, third one initiation of polypeptide chain, fourth one being elongation of the polypeptide chain, and polypeptide chain termination is the final step. Let us now see the first step, activation of amino acids. Amino acids in the cytoplasm will be in dormant stage, so they cannot take part in protein synthesis. Hence, they have to be activated by giving them energy. The activation is brought about by the addition of ATP, whose molecules unite with amino acids to form highly reactive amino acid phosphate adenyl complexes, which are known as amino acyl adenylates. The process of activation is brought about by specific enzymes. Usually, each amino acid has its own specific enzyme. So, there will be as many enzymes as the number of amino acids. Each enzyme has double specificity as it recognizes its own amino acid that finds out its own mRNA. So, we can see here the equational form as to how the amino acids are activated in the presence of the enzyme. So, amino acid, it requires some energy which is provided by the ATP in the presence of amino acyl synthetase, the enzyme. It forms amino acyl adenylate phosphate, and pyrophosphate is also released. So, the second step being the transfer of the activated amino acids to the tRNA. The activated amino acids, which are now bound with the enzyme, are called amino acyl adenylates, which get attached with their tRNA molecules. This attachment is catalyzed by the enzyme amino acyl tRNA synthetase. This results in the formation of amino acyl tRNA complex. Now, the initiation of the polypeptide chain begins. Once the amino acids that are present in the cytoplasm are activated, now the chain formation begins with the initiation of the polypeptide chain. Initiation of the polypeptide chain is brought about by the amino acid methionine, which is coded by AUG codon. AUG is also called as the initiation codon. In E. coli, 2 different tRNAs receive the methionine. tRNA M-met, which is the non-formulatable tRNA, and the second one is the tRNA F-met. F indicates formulatable tRNA. tRNA F-met deposits formulated methionine as the first amino acid of the polypeptide chain, and tRNA M-met deposits methionine as the first amino acid of the polypeptide chain, and thus initiate the formation of the polypeptide chain. initiation of the polypeptide chain in prokaryotes. In prokaryotes, initiation takes place by the formulated methionine formation. So, in E. coli, formulated methionine is picked up by a different tRNA which is designated by tRNA F-met, that is formulated tRNA. Methionine is at the intercalary position in polypeptide chain and is deposited by another tRNA, tRNA M-met, that is non-formulatable tRNA. The initiation codon AUG initiates the amino acid N-formyl methionine in case of prokaryotes. The amino acid tRNA complex is attached to the initiation codon AUG on mRNA by its anticodon UAC in the presence of the initiation factors IF2 and IF3 plus GTP is the energy source to form the initiation complex. In case of initiation in case of the eukaryotes, it takes place or is brought about by a special MET tRNA, but the methionine is non-formulated because tRNA F-met is absent in plants and the enzyme transformylase is absent in case of animals. There is another tRNA for the placement of methionine inside the chain, GTP and 3 protein factors EIF2, EIF2 prime, EIF2A2, EIF2A3 and EIF3 are loosely associated with a small ribosomal subunit and they are involved in the initiation of the polypeptide chain formation. The largest subunit 50S of ribosomes attached to the 30S initiation complex to form complete initiation complex 70S. The larger subunit has 2 binding sites for tRNA, that is A or the acceptor site and a P or peptidyl site. So, the elongation of the polypeptide chain begins with the complete formation or union of both the smaller and the larger subunits of the ribosome. The formation of the 70S mRNA F-met tRNA F-met complex and the elongation of the polypeptide chain occur by the regular addition of the amino acids. The elongation begins with the binding of the amino acid tRNA at the site A, that is the acceptor site of the larger subunit of ribosome, which has 2 slots, P or the peptidyl site or donor site and A site, acceptor site or amino acyl site. The amino acyl tRNA complex is attached to the acceptor site and tRNA carrying the peptide chain is attached to the peptidyl or donor site. This process involves GTP to provide necessary energy and the second amino acyl tRNA attaches to the A site and binds to the second codon on mRNA. The above step is followed by the formation of peptide bond between the carboxyl group of the peptidyl tRNA at P site and the amine group of the amino acyl tRNA of site A. With the formation of the peptide bond, the tRNA from P site is released and the polypeptide chain is transferred to tRNA present on A site. Once the tRNA from P site is released, the peptidyl tRNA from A site shifts back to the P site. The whole process is aided by a molecule of GTP and transfer factor enzyme translocase. During the process of elongation, the ribosome shifts along mRNA in 5' to 3' direction so that next codon on mRNA will be available at A site, which requires a G factor and GTP. With the movement of one ribosome along the length of mRNA, the initiation point on mRNA becomes free. It can form an initiation complex with 30S subunit of another ribosome. In this way, a number of ribosomes get attached to a single mRNA molecule. This complex is known as polyribosome complex. During the process of protein synthesis, a number of ribosomes can be seen attached to a single mRNA molecule at a time, each with a polypeptide chain under formation. The size of polypeptide chain on different ribosomes being different. Elongation of peptide chain involves elongation factors, elongation factor TU, EFTS, EFG in case of prokaryotes, and EF1 and EF2 in case of eukaryotes. Now, once the chain has elongated and the required protein has been synthesized, the chain has to be terminated. So, the termination of polypeptide chain occurs when the ribosome carrying the peptidyl tRNA reaches the termination codon UAA, UAG and UGA present on mRNA at the end of each cistron. Releasing factor R1 and R2 are needed for chain termination. R1 is specific for UAG and R2 is specific for UGA. The releasing factor with the termination codon forms a complex which induces the enzyme peptidyl transferase to catalyze the termination and release of the polypeptide chain. The free ribosome now dissociates into two subunits with the help of dissociation factor F3. Please like, share and subscribe so that it would be an encouragement for me to make many more such videos. Thank you for watching.